African swine fever is a viral disease that is highly contagious for both domestic and wild pigs, with mortality rates approaching 100%. This disease can have heavy consequences on both pork and protein supplies. The disease historically has mostly remained endemic to sub-Saharan Africa here in 2005. African swine fever was not thought to be a threat to the rest of the world, as this disease was located mostly in Africa. However, this changed in 2007 with a single introduction into the Republic of Georgia, which had rapidly spread to surrounding countries in the European continent, and by 2016 had spread to the north of Russia and all the way west into Poland, where African swine fever outbreaks continue to occur in these areas. In 2018, ASF was introduced into China, the largest swine producer in the world, producing over 50% of the world's pork supply. The introduction into China caused a rapid spread to neighboring Asian countries, and by 2021, the disease had spread from China to 15 countries in Southeast Asia. Recently, ASF continues to spread on the European continent and entered Germany in 2020, and it's continued to spread in Asia into Malaysia in 2021. The disease has now reached pandemic proportions, resulting in what had been estimated by the Food and Agriculture Organization of the United Nations that the loss of half the world's pig population occurred in 2019. The introduction in Bulgaria caused a 25% swine inventory decrease, in China 50%, 25% in the Philippines, and 45% in Vietnam. These significant decreases caused a threat to the stability of the global swine industry and the world's protein supply. A few weeks ago, the disease had been detected in Dominican Republic after 50 years of being absent in the Americas. This is the first outbreak in recent history of ASF in the Western Hemisphere. As such, the potential introduction of ASF in the United States remains the #1 concern of the U.S. pork industry, which ranks as the third largest swine producer worldwide, after Europe and China. Now in 2021, African swine fever is a worldwide problem.
The lack of commercial vaccine is one of the main factors allowing the rapid spread of the disease worldwide. Researchers across the globe have tried unsuccessfully in the last 50 years to develop a safe and efficacious African swine fever vaccine. The challenges include the complexity of the virus itself, with one of the largest viral genomes known, and over 50% of the genes uncharacterized with no known functions. Additional factors contributing to this failure are the lack of an identification of the viral antigens responsible on the induction of the protection, as well as the immune mechanism mediating that protection. Over the years, attempts were made to develop vaccines from apparently attenuated field strains. However, these strains, although less virulent, resulted in unsafe vaccines that over time caused symptoms associated with that African swine fever. These vaccine strains had the potential to spread in big populations and were harder to test, hampering disease control efforts. Today, similar attempts have resulted in the illicit use of unapproved African swine fever vaccines in China, resulting in vaccine strains with residual virulence that are spreading and causing new disease outbreaks, further enhancing the African swine fever pandemic.
In response to the ASF disease pandemic, ARS scientists at Plum Island Animal Disease Center started in 2008 a new ASF research program focused on the development of a safe and effective vaccine. After characterizing the African swine fever Georgia strain and several years of ARS laboratory experimentation, they were successful in optimizing a complex process that allowed the elimination of specific viral genes associated with virulence, along with the efficient process for purifying the resulting attenuated vaccine virus. Using this complex process, first ARS scientists had to identify genetic targets, and they created a custom computational pipeline that takes into account different sources of bioinformatics and functional genomic data. After identifying these targets, ARS scientists engineered several ASF gene-deleted viruses, resulting in the discovery of several viable ASF vaccine candidates, one of which, based on the deletion of the I177L gene, was determined to be the most promising vaccine candidate, based on the results of a proof of concept clinical studies in pigs. This discovery from 2019 was granted a US patent in May 2021. What differentiated the I177L vaccine from all other vaccine candidates included demonstrated efficacy with a low dose, sterile immunity, meaning no virulent virus could be detected in any of the tissues examined in infected pigs after challenge, absence of residual virulence, and the ability to grow the virus to high titers in primary macrophage cultures, which are used for vaccine production. This vaccine has also shown the ability to be an effective vaccine when given either intramuscularly or by the oral nasal route, opening the possibility to deliver the vaccine to wild boars by a bait system. ARS scientists successfully completed the needed clinical safety studies needed to obtain exclusion for the I177L vaccine from the Select Agent Program, which is an important first step in the transfer of the vaccine to commercial partners for further development, as this would allow the vaccine to be produced in a BSL-2 facility without the oversight of the Select Agent Office.
The news of the I177L vaccine invention prompted the government of Vietnam, who was suffering almost a complete loss of their swine production to ASF, to invite ARS scientists to Vietnam to explore a potential partnership to develop, manufacture, and commercialize the I177L vaccine candidate. ARS scientists traveled to Vietnam on February 2020 to meet with the Vietnamese official from the Ministry of Agriculture and potential collaborators from the Vietnamese pharmaceutical industry. This visit was critical and resulted in the successful identification of a vaccine company, Nabetco, with the needed qualifications to commercialize an African swine fever vaccine. Upon our return to the United States, we successfully worked with Nabetco to develop a vaccine development plan and helped the company navigate the USDA technology transfer process, resulting in ARS issuing Nabetco a license agreement and a collaborative material transfer research agreement on August 2020, with the materials being successfully shipped to Nabetco on September 2020.
Upon receipt of the I177L vaccine, we in Nabetco immediately started having remote discussions by Zoom, as travel was impeded by COVID-19. A few months later, the first successful commercially produced batches of the I177L vaccine and primary swine macrophages was completed.
This was followed by the execution of a full vaccine development plan, consisting of critical safety and efficacy clinical studies that were submitted to Vietnamese regulatory authorities in record time by February 2021, only 5 months after receiving the seed vaccine from ARS.
The success and speed with which the vaccine development plan was executed was due to an effective collaboration and trust established between us and Nabetco, achieved through regular meetings late at night due to the time difference between the US and Vietnam. This collaboration resulted in a successful development plan that showed that under commercial production, the I177L vaccine candidate was highly effective both in commercial and local breeds of swine.
Important next steps to follow will be the massive production of the vaccine and their assessment in controlled field conditions, and, if successful, the commercialization in other countries of Asia affected by African swine fever.
Also, and importantly, the stockpiling of the vaccine in the US so it can be rapidly deployed should we ever have to face the devastating effects of an African swine fever outbreak in this country.
Further, as the use of vaccine in ASF endemic countries becomes widespread, the threat of African swine fever for US pork producers will be greatly reduced, and the reliability of a critical food staple across the world will stabilize.